Record from joined online as he is in US right now. I get Rahul Singhi, Director Poornima Group, Dr. Bhaggat Devla, Dr. Gautam.
हम कई बार हिचक की वजह से नेटवर्किंग नहीं कर पाते, बट इसका गुरु मंत्र ये है कि अननोइंगली नेटवर्किंग कर लो। तो डे वन पे ही अगर वो अननोइंग नेटवर्किंग हो जाती है तो आप एक नेटवर्क में आ गए, एंड देन इट इज अबाउट डेटा शेयरिंग व्हिच विल हैपन ओवर द इयर्स। इट विल नॉट हैपन ओनली इन दीस 5 days.
Of computational biology is to provide computational solutions to biological problems. For example, it explores the hidden biological information and interpreting some meaningful thing using IT. So, biological principles are not obvious, not physically you can't see the cell. Talk about cell, our eye cannot resolve to see the cell. How come you see DNA, protein? These are the subcellular. They are present in subcellular localization, subcellular compartment. Talk about cell organelles. The size is so small. We use electromagnetic. Nothing but physical. Again, it's mathematics. But now I will definitely discuss about those where we have reached. Right? I said hidden biological information. For example, structure of a protein. Just mere photographic X-ray image of photographic image. We have developed a very beautiful three-dimensional. And if you look at the raw data, it's nothing but just spots, crystallographic spots. But what we have achieved is the structure of the protein, and that is most important part in biological sciences, biological process to run our life, to regulate our life. We talk about enzymes, we talk about anything, protein, enzyme, anything is basically depends upon the shape, its configuration, structure. So structure of this protein is key, is critical to know the function. And we have solved through crystallography. We have solved through, but for last many years, as I discussed in my morning address, that for last 40 years, we have been trying and keep trying. We have limited, restricted, experimentally, either crystallography or NMR. But creating a crystal of all biological objects is not possible. It's a big limitation to create a crystal. Until unless you get a crystal of a protein, you will not be able to solve it. So getting a crystal, again is a, crystallography is again completely mathematics, space group, symmetry, lattice, you work for symmetry in space group, try to look for, then you create an orthogonal matrix, you create a hypothetical virtual matrix. You put a virtual hypothetical matrix over an object which solve the real protein problem. But you create a virtual matrix and you try to you arrange that matrix so that one lattice contains a unit cell, the lattice cell contains one protein molecule. This is what your objective is, ultimate objective is. To arrange that virtual matrix so that you can. So, but ultimate result is structure of the protein. What you got is structure of the protein. So, biological data, we talk about RNA or a DNA or a protein. So this is a biological data that we are working upon. We are working upon the DNA, we are working upon the RNA, we are working upon the protein molecule. This is our input. But this input has again a format, some sort of a format. We call file format. Then we use informatics and we read the sequence data, the pattern, the call, base call in triplets, and based upon we develop a consensus DNA sequence. And then this DNA sequence can be stored inside the computers in normal text format. You store your DNA sequence in text form. Then you can work upon this. Once you store the information inside the computer, that means you now have a string of characters to play upon. You can identify pattern, you can identify composition. Now you can filter it out depending upon the data. You compare multiple sequences. You try to establish evolutionary relationship between sequences. We call phylogenetics, molecular phylogenetics, where we try to study evolutionary sequences, how sequence evolved, evolutionary pattern, how evolution occurred, how sequences started vary, how they started accumulating this variation we call mutation, and this variation keep on accumulating, and after certain time frame, evolutionary time frame, one gene becomes two different genes because of those accumulated variations. Maybe 500 years before there was only single, but with the time, with environmental pressure, with our ecosystem, they start accumulating changes in the DNA, and after 500 years, if you look, then we see a divergence, we see a differences in those two sequences, and those variations accumulated naturally, depends upon our environmental condition, because we have a very robust inbound system where we call this a state of hemostasis, and this state is very conserved. Can't go beyond that. You have limitation of a temperature, so you know your comfort temperature, your pH, your blood pH is again very important parameter. So your body has its own internal environment. You can't change the environment, otherwise you will die. Every organism has its own. We know organisms, but how do we define life? Any object can be referred to as a life. We create objects. In programs so simple, in your those who are from computer science make create an object life, and you know define the attributes, make a constructor, that's object, create an object of life. Then you define the methods of object, can move, can bark, can run, can laugh, can then create a structure, maybe like if you talk about structure, physical structure, 4 hand, 2 hand, vertical, horizontal, define object, life, but it's a dynamic. It's not static, it's dynamic. In physics, we talk about law of conservation of energy. We talk about the enthalpy and entropy. We know the entropy of the universe is continuously deteriorating. It changes from ordered state. It is our tendency to go towards unordered state. But if we want to restore to our native state, we need to put energy, we need to put time, we need to put effort, then only we can be arranged again. But after 1 week, we become again scattered. It is naturally to be disordered. Disorderliness of the system is natural. But to become ordered, we need to put energy to become in ordered state.
Thereafter, I shifted over to industry. I have done medical writing for firms like Takeda, Stryker, and Walter Kluwer's. And thereafter, I met one day. I was working with Takeda, and I pointed out certain things in the clinical trial going on at ILBS, Institute of Liver and Biliary Sciences. So I met Dr. Sareen, and Dr. Sareen said that I am working on a project which is an excellent project, which is of Indian and Norwegian government through Ministry of External Affairs. There is a project. So we try to develop a vaccine for liver cancer. So it is an excellent project. So I met Dr. Sareen, and he hired me as a clinical research associate at IIT. So I worked for around 3-4 years there. And we try to develop a exolive vaccine. So I briefly tell you about what the vaccine is all about. So we are working on the exosomes. So what are exosomes? Exosomes are extracellular vesicles. So the extracellular vesicles are small cells of micronuclear size. They are being expelled from all cellular bodies. So what we are using them to package our medicine, which is doxorubicin. Otherwise, it is being resistant. Cancer cells are being resistant to doxorubicin, but we are doing a strategic engineering with that. We are packaging that doxorubicin in the exosomes, and then we are delivering it to mice. So we did experimentations on mice. So if it got cleared, so this was my work. So it requires certain molecular work, aptamers. So then COVID came, and the things that were being delivered from Norway, they are being halted. So all the things got disturbed, and then the government funds also. So we face several hurdles. I was telling this Dr. Shilpi that we are faced. That is why we are having so much talent in India, and of course in your institute as well. So these are the hardships we face, and we will be discussing how can we overcome them and how can we go forward. So I try to give you an idea about what the mathematical modeling. You are all experts of mathematics. So see, now are the days where we need to collaborate. So nothing, an individual, whether it's me or whether it's you, we are not going to be making anything productive or useful for the society unless we collaborate. And we are very good, I mean all of Indians we are very good in individual work. We do very sincerely, but when it comes to teamwork, we somehow till होता है ना सबके साथ में. तो यही पे ये है कि वो काम तभी आगे जाएगा जब हम टीम वर्क में काम करेंगे. So आज की यही है कि I will be discussing mathematical modeling for personalized cancer treatment strategy. Mathematical modeling, you guys know better than me. I will be talking what we need from you. So what a scientist, because this is the frontline area. This is in the frontline area of research in the ATAL program. It was given. So I took that heading, and I am going to talk about it. So what is a personalized cancer treatment strategy? So cancer is highly heterogeneous. So by heterogeneity, we mean that one cancer is different from other cancer. For example, a cancer called adenocarcinoma. It is very, very aggressive, and for 3-4 months, it is very difficult to diagnose also, because it happens in the rim, at the rim of the, where the connectivity of pancreas and intestine. So at that place, it is very difficult to diagnose that cancer, and treatment is very difficult. Again, we are using biopsy sample, we are using FIF, fine needle aspiration. So in which we have to pull out the tissue. From using a fine needle, we need to pull out in order to make a confirmatory test for cancer. All the cancer diagnosis tests are doing that. It is being given by the Mayo Clinic also. So if you are doing FIF, for cancers like adenocarcinoma, it is going to be doing tumor seeding. So अगर आपने उसको fine needle aspiration यूज़ करके आपने उसको निकाला, तो वो ऑलरेडी बहुत ज़्यादा एग्रेसिव है वो ट्यूमर, तो वो और भी जल्दी से फैलेगा. तो मेयो क्लिनिक के गाइडलाइंस हैं, बट हमारे पास और कोई डायग्नोसिस के इतने ज़्यादा कंफर्मेटरी टेस्ट नहीं है, तो हम लोग वही यूज़ करते हैं, फाइन नीडल एस्पिरेशन. सो आज का वाला टॉक है कि अभी किस तरह से हम एक सिम्युलेशन मॉडल बना सकते हैं, सो दैट वी कैन ट्राई that how can our therapy work on those models. तो अगर हमारा simulation model work करेगा तो वो therapy भी work करेगी, क्योंकि हमारे पास results हैं. We have lots of patient data. Sorry. So what are our strengths? इंडिया के पास बहुत सारा पेशेंट का डेटा है जो कि वेस्टर्न कंट्रीज या वेस्टर्न प्लेयर्स आर लुकिंग फॉर इट. व्हाई दे कम टू इंडिया? दे आर लुकिंग फॉर द पेशेंट डेटा. दे आर वेरी डेटा हंग्री पीपल. So we are having patient data, so we can make a simulation model using that patient data, and we can combine the biology, treatment and mathematics. So now, what is a personalized medicine? So one medicine is not appropriate for everyone. For example, I say a drug called TP-Lumab. It is not very useful for one person. It might be useful for the other person. So we cannot generalize when it comes to diseases like cancer. We cannot give one single medicine to all. It might give good results to one person, the other person the result. So how can we diagnose that this is the person that is fit for simulation model? And this approach is called a personalized medicine. Have you ever heard of Dakar Declaration? It is between French government and African countries. It is about, since we study that the evolution took forward from Africa. So French government का और African government का एक personalized medicine को लेके एक declaration है which is called Dakar Declaration. And it is about the personalized medicine जो वो उनको दे रहे हैं. So हर person की अपनी unique requirement है, उसके हिसाब से उसको treatment देना, तो उसको बोलते हैं personalized medicine. Okay? So how precision oncology use patient specific data. So for example, certain set of parameters for me might look like certain parameters for other persons. So there will be a resemblance. So we are having data of patient that has passed, yeah, the patient that are sustaining, or there is a data. We can classify the data. So जो अच्छे results जिनके आ रहे हैं, we can simulate that data for that person. So this is how the simulation model work. तो simulation model बनाना ये mathematicians का काम है. मेरा काम है आपको सजेस्ट करना, आपको बताना कि हाउ आपका स्कोप क्या क्या है. आपको कोलैबोरेट करके आप पेपर्स लिख सकते हो, आप सिमुलेशन मॉडल बना सकते हो, और किस दिशा में आपको बनाना है फॉर दीज कैंसर्स दैट आर वेरी वेरी डिफिकल्ट, जिसमें कि फॉर एग्जांपल एडिनोकार्सिनोमा, जिसमें कि 3-4 महीने बिना डायग्नोसिस के पेशेंट्स मर जाते हैं. ठीक है, दिस इज़ वेरी बहुत सारे मेरे नोन लोगों में भी हुआ है, और कुछ मतलब ऐसे भी हैं जो कि जिनके ड्रग आई है इस कैंसर के लिए. फॉर एग्जांपल आई विल बी डिस्कसिंग अ पेपर ऑल्सो, ज़ैनिमेटा मैप करके उसको FDA का फास्टेस्ट रूट से अप्रूवल मिला है, लेकिन इट इज़ वेरी एक्सपेंसिव. अभी बहुत सारे लोग उसको रीच नहीं कर पाएंगे, और इंडिया में अभी वो आई भी नहीं. तो आप बहुत अच्छा एक मॉडल बना सकते हैं फॉर प्रिसिशन मेडिसिन, फॉर पर्सनलाइज्ड मेडिसिन. ओके. Biomarkers, see, there are certain agents like CCA. So about that present of minutest amount. So मैंने आपसे अभी कहा था कि फाइन नेडल एस्पिरेशन से हम लोग कंफर्मेटरी टेस्ट करते हैं कैंसर का. तो अगर हम कोई ऐसे बायोमार्कर्स का टेस्ट कर लें जो कि कंफर्मेटरी टेस्ट हो जाए, तो हमें फाइन नेडल एस्पिरेशन करने की ज़रूरत नहीं पड़ेगी, और वो बायोमार्कर्स भी हमें कंफर्मेटरी टेस्ट दे देंगे. बट दिस इज़ अ स्टैंडर्ड. आप कोई भी पूछेंगे हॉस्पिटल में तो वो बायोप्सी करते हैं. तो बायोप्सी कई बार क्या होता है कि वो ट्यूमर CD करा देता है, और स्पेशली जो कि ऐसे कैंसर्स हैं जो कि बहुत ज़्यादा एग्रेसिव है. ठीक है, सो दिस इज. आप कोई भी क्वेश्चन पूछ सकते हैं. आई वुड लव टू बी मेकिंग अ सेशन इंटरैक्टिव. आपकी जो भी कोई क्यूरियोसिटी है वो भी आप मुझे पूछकर रोक सकते हैं. ओके.
Of Pennsylvania, USA. On his return to India, he served as a Ramanujan Fellow and later as Assistant Professor at IIT Indore, and is now an Associate Professor. His pioneering research spanned microbiology, cancer biology, and host pathogen interaction. For his outstanding contributions, he has been honored as a Fellow of the Royal Society of Biology UK, We are truly privileged to have him with us today. So please join me in extending a warm and hearty welcome to Dr. Hemchandra Jha.
I was also affected SARS-CoV-2 and get the some symptoms early as a biologist, so we started medication early and then went for the real-time PCR that almost all of us went, as well as the CT scan also. So when I went for the CT scan, it was showing 32% glass of PCE, like lung infected. And as I medicine started 2-3 days ago, so after 2-3 days of this 30% infectivity, I feel I'm okay. So I got that question, why 32% is too high? And then a lot of reports also coming from some people that yes, someone have 80%, someone have 50%, but that looks very unmatching. Like someone is really infected, someone may not that much. But these numbers are not that good. So then I spoke with some radiologists as well as some doctors that how we are deciding this percentage. And they told us that there is no decimal pointing in the, because this is lung, right? 3 and 2, 5 lobes. So they are giving like 0, 1, 2, 3, 4. And there is no 0.1, 0.9, 0.8, 0.7. But as you know, this is the area, right? So it has to be some, it cannot be 1. But that is no, that classification is not exist till now in the medical system. And as per the, this is the endemic, so WHO guideline, even you have 0.1% infected, you have to give a score 1. So 1, 1, 1, 1, 1, it is 5, right? And into percentage like 4, so it is 20%. Although if it is less than 5%, like if it is 0.1, so 0.1, 0.1, 0.1, 0.1, 0.1, 0.5 into 4, 2%. So less than 5%, there is nothing. But in this case, because of the guideline, or you can say it's not that much, it's not decimal by the punctual, it is coming to the 20%. So now, when doctors have and you have the, your 20% lung infected, everything is changed. So our first question is that, and then we collaborated with the Department of Mathematics, the people that are doing the vector support machine as well as the AI. So we utilize that, and then we go for the another restore also. So before that, I am going to something about our lab. So my infection group is infection biology group, where last 9 years we are working. One of the virus, Poornima virus. This is the first virus known as in 1964 known as the causing cancer. So first is called tumor virus. Cancer is tumor, right? So first known tumor virus that is we are working in one of the aspects. Second thing, we are working in neurological modality because various these infections are causing the Alzheimer, Parkinson, multiple sclerosis, that as well. Third thing that we are covering gastric cancer. Many of us have gastritis, gastric problems, right? But then when this bacteria having the gastric problem with alone, but when this co-infection is there, it is very high. So we are trying to understand why it is so high, why it is so difficult to treat. So those factors we are trying to understand. And now machine learning approach that I just discussed you and going to explain in more detail. Low-wide GGO, ground glass opacity detection, where not only the COVID pneumonia detection, we also try to correlate whatever the lab data is coming, like for biochemically or inflammatory markers, like from those days 20-30 markers, GGT, all these lung parameters, blood parameters. So is that something can be associated with that we also try to understand it. And then various proteins of SARS-CoV-2 that we are understanding. And also these days the gut is also called what? Secondary brain. Earlier also we got to know that you need to keep your gut healthy, but it is now heavily evident that there are a lot of things, anxiety, depression, mood swing, even diseases are also not only contributed at the brain, a lot of things have contributed at the gut also. So a lot of secretions and direct connections like vagus nerves or enterocytes that is directly transported. Even now evident came that deposition, what we are telling in the Alzheimer as well as the Parkinson, that is also making many times in the gut and simply transported to the brain. So that also we try to understand it. So they have some overview of the lab, and this is some of the aspects that in the COVID-19 we covered. Like initially we all are in the home because everywhere is curfew. So we and our India, fortunately the wave 1 there is no much data as well as no much patients also. But in France, Italy, Netherlands, Spain, Sweden, Iran, as well as some other countries, they have a lot of infected, like 10 to 50% people infected. So we simply tracked the data from their health ministry and try to understand exactly why so much death in some countries are occurring and what are the reasons. So based on that we got some comorbidity, some medicines. There are some countries are using some medicines, some countries are using another medicine. So is that medication has some issue or some earlier diseases that is have some issue, so that will be published that paper. First thing, and the second thing, that in the same time we collaborated in the Kalinga Institute of Medical Sciences, Odisha. That is the one of the hospitals. They had 2,000 beds for the COVID. So they are giving us the 2,000 patient sample data. So all these COVID people data. So they have like people with the earlier cancer also. So different kind of cancer people, if they got the this COVID, then what could be their medication and what could be their biochemical parameters, like malaria people infected and then the COVID, or if they have encephalitis, meningitis, or some other neurological problem and then they are getting COVID. So those kind of a study also we have done, case study as well as series, and mutation study. We all know that Wuhan, whatever virus came, after that one mutation 6 months and then another mutation, and at last this delta mutation, the second wave, and so deleterious. So exactly what kind of things are changed and what are their consequences, so that will be included, and this extended part I will talk in more detail with data viruses. That is the in second wave we had problems. And then therapeutic, what could be the, if we target the virus proteins or our own host proteins, what could be the possible approach, as well as some of the infection through infection. So some of the study we published last few years. This is my research group. Here, those are in the red circle. They are already passed out PhDs. Right now, some people are here. So we are, I mentioned that we are working in the several aspects like cancer, neurological issues, as well as the gut-brain axis. These are the mostly in the COVID. Now coming to the first part, redefining the low-wide ground glass of PCE in COVID-19 through deep learning and its correlation with biochemical parameters. So first tell me, how many of you have mathematics background? All of you? No. Other people have? CS. Okay. CS. So lot of people. So a lot of people in our institute also, either CS or the mathematics, they are doing health related stuff, and they are really doing good. But then I always try to convince them, not able to do it yet, right, that getting the samples from US or Europe or Australia or XYZ countries and then developing algorithm is good. But it is not going to solve actual problems because we have different set of genomics, not exactly same, different set of problems. So yes, if totally dependent on these data, it is difficult. But try to collaborate with some experimentalists who are generating the data, and then you utilize your algorithm. Possibly in the long run, that is only going to help. So that is my personal opinion. I try to convince you. So here what we started, when we find out that yes, so what AI ML do in the healthcare? There could be many things. Like I think many of you are already doing this.
At the same time, we are not understand the limitation of this device, then it creates the hell of the life. And in these days, this is going on. Why I am talking? Because my topic and talk little bit related to this area, right? Not device. I am talking about the computer, right? So my today's talk, उसको email करें, उसको explain. also pay my gratitude to the AICT ATAL to providing the opportunity and financial support to the organizing committee member and as well as a being of resource person to give me opportunity to talk on the recent trends of mathematical and computational biology. Can anyone, it is an interactive session, so can anyone tell me what is the role of the mathematics in computational biology? Computational is a different thing, biology is a different thing, and mathematics is a different thing. Anyone? What is the connection? आप गलत भी बोल सकते हैं क्योंकि आप मेरे को लिख सकते हैं, right? और मैं भी कुछ सही और गलत बोल सकता हूँ, right? So anyone, please tell me, do you understand what is the connection between the mathematics, computer, and biology? What is the connection? Okay, madam, what is your good name? So Lakshmi is okay? Yeah. So Lakshmi is tell about the, if we have some kind of the analysis, we convert in the form of the mathematical model, and then we have analysis with the help of the some of the computational techniques. That's why it's a connected, yes? Moderately it is a correct. But if we are talking about a single person, if I am talking about the myself, right? Then every kind of analysis is not able to predict this particular analysis for the another person, right? So that's why the mathematics is giving the some kind of the opportunity for the engineers and scientists. What is the benefit? If we understand the particular analysis of the one human, then we create a one particular model. I give the one very simple example. If you want to understand the movement of Mike from this distance to this distance, then I create a one differential equation, right? And with the help of this, in this particular distance, we can observe the understanding of the behavior of the another person, like a Gauravji, like a Prakashji, like a Saketji. We can, right? So this is the advantage of the mathematics. Okay. Now we are talking about the computation. If I am talking about this particular space, then mathematical model. But if we are talking about, we have some many data, some of the distance, like a similar kind of the distance and similar kind of the moment in another place, or maybe the multiple places, right? So we compile all the data in a particular model, then we can analysis. So then computer is involved, right? Then this analysis is able to do with the help of the computer. Okay. Now I am talking about the biology. In these days, the problem we saw after the pandemic. What is the problem is going on in these days? We saw and we heard some of the celebrities. Normal human being is died, no one knows. But if a celebrity is died, everyone knows and everyone are discussing. We heard many death, recent death. Those are having the age of 35 or maybe between the 55, 35 to 55. Okay, not an older person. This is the age in which after the pandemic in 2023 until now, many deaths are occurred. Some of the good cardiologists, they are very good cardiologists. They are helping to the humans to survive in the life. But suddenly we heard about that this particular cardiologist is died after the OPD. Why? Big question. The second, we heard about the many sportsmen. Those are very very good. Bollywood, Hollywood actors, actresses, they are very good, health conscious. But suddenly they die. Why? What do you think? What happened is going on inside the body? Why they are dying? There is no pandemic right now, but they are dying. And the diagnosis is very strange. I think yesterday Professor Jha is taking the session. He is my good friend from the IIT Indore. So he is also working in the biology field, right? So can you tell me why it is happening? And ऐसा क्यों हो रहा है? Why? Why? But body को मालूम नहीं चल रहा है ना? If someone is the Hollywood, for example, I am Amitabh Bachchan. Okay, you assume I am not looking like that, but you assume that I am Amitabh Bachchan. Okay, so I have a dietitian, I have a doctor. Every day someone is monitored to me, but suddenly I die. Why?
The vaccine, most probably what we have, the vaccine that we have taken it was RNA-based vaccine.
Yeah.
So we have done some modifications in our DNA or RNA thing what we are doing.
Perfect. What's your good name, sir?
Dr.
अमित जी, बहुत ही बढ़िया क्लैपिंग फॉर द अमित जी। giving the exactly correct answer which I am giving in my presentation. We have having inside the body some vaccinations, different names, right? But after the vaccinations, the vaccine is create some kind of the, the neurons, right? The neurons, sir is mentioning that the DNA, right? The DNA is directly impacted by the neurons, right? Neurons होते हैं हमारी body के अंदर बहुत सारे. Neurons is changing their structure, right? That's why the DNA structure is going into the change, and that's why the doctor is not understanding कि suddenly the cardiac arrest is happen in some human body, but before that there is no symptoms. This is the problem, and this is the danger alarm for all human people of the around the globe. It is not a study point of the India. We are very populated, but it is a very, very critical situation around the globe. We all are having the vaccination, and now this is create a problem, right? Now today, my talk is basically the study of the mathematical analysis of infectious disease by fractional calculus. Do you know the fractional calculus? If we are talking about the movement, then we know the differential equation. But if we are talking about the fraction of seconds, my movement, then we require the fractional, right? Simple differential equation giving the differentiation, right? In some kind of the second, some kind of the.
Convey my thanks to Poornima College of Engineering, Shilpi ma'am, Nikita ma'am, and director sirs to invite me and share some research topics with this faculty members and other participants of this FDP. So, I think so. Is it from here? Is it fine? So basically, we just say first of all, this topic is mathematical modeling and AI for renewable energy resources. Might be some of the participants is computer science background, others from electronics. But I think so. Both terms you are familiar with AI as well as with renewable energy sources. Just renewable energy resources, basically just like solar, wind. Right now we are focusing on these non-conventional energy sources because day by day there is a depletion of conventional energy sources, just like your diesel, coal. They are depleting, and even they will produce hazards to the environment. So that's why we have some planning and even governments and throughout the globe we are looking for alternative options, and right now the research is going on this particular domain. And if we talk about this electrical domain, then definitely solar, wind is there, but along with that electrical vehicles is also there. So these are some outlines of these presentations. Right now we have taken that paper I will discuss here, this estimation of calorific value using artificial network based on stochastic ultimate analysis. And similarly, the next topic is also related with this one, and this is the efficient high heating values. Again, this is the calorific value. What it means, calorific value means to say, if you talk about this biofuels, then they have some calorific value, a high heating value. that will shows how much energy can be generated from any resources. So that's why we are here considering MSW, that is municipal solid waste. This is interdisciplinary research, basically with the civil engineering department. I have done some work with the other faculty member, Disha Thakur, that is also one faculty member there in our branch, in our UIT. So the outlines of this is artificial intelligence, I think so now you are familiar to what videos are there. So what is artificial intelligence? And then introduction waste to energy, how waste can be converted into energy. Again, there is specific term, again calorific value, high heating value. Fine. So after that, what methodology we have adopted to carry out this research, and I think so we have taken 1 year to carry out this research. For there are 2 publications from this particular research area. Then implementation, result and conclusion, and what about the future scope? What are different future scope for in this area we can go. Okay. Along with this area, you can also implement the same research in same field, in other field. Okay. Fine. So even this work that we have concluded in second paper, that is also a work carried in electrical engineering domain. This is a similar type of work. We have estimated the solar radiations, forecasting of solar radiation, estimation of solar radiations through this methodology we have opted. So if we say just generalize about the AI and human intelligence, what is human intelligence and what is artificial intelligence? So everyone know that AI processes data using some algorithms and make objective and data-driven approach. And on the basis of that, we comes out some decision. What is the different decision? On the basis of that, we decide how it will be performed. But ultimately, the main objective is there is the data approach. Means to say data, we require the data. If we go for to make any machine intelligent, we require the data. But what the issues with the artificial intelligence? Humans combine emotional and logical reasoning, adding depth to their decision making. Everyone we learn the things. Even a child starts to learn from his childhood and then grow day by day. On the basis of that, they learn the things. Initially, if I take the example of a car, then definitely if you start driving, then everyone see accelerator, first gear, just like that, we notice that. But at the end of the day, means to say with that time, if you are fully trained, then you are talking with your phone or with the phone or even in car or on speakers. But your mind is so trained that your accelerator and gear shifting is done automatically. You are not so much concentrated on that. Means to say, training is good. Learning is good, training is good. So similar type of work, means to say machine can do. But what is the main reason that machines lack? Main reason is emotional thinking. Means to say if you train some machine with help of some particular data, then it will perform only that particular task. Not think about its good, its bad. But you start your thinking, means to say you will think if this is right or this is wrong. This is the basic difference between human intelligence and artificial intelligence. So similarly, learning efficiency, AI requires large data set, but human beings can learn through some examples, and on the basis of that examples, he can do another task also, perform on the basis of their understandings. Similarly, adaptability, human easily generalize knowledge across the task, you can make some generalizations. But for AI, we have to make that generalized.
Having one rod, was measured between the 2 points of a rod, listening, reading rod. Now, anyone is having a new definition what they are like? How they are defining 1 meter or anything else? So what they are doing now, they have initially they were physical artifacts for 1 meter, all these things, 1 meter, 1 kg. Time was related to the superfine like oscillations for the cesium atom. That was already related to natural constant. But now these things also have been related to natural constant, because natural constants will not change. If artifact is there, that will change. It may corrode also. Or if 1 kg is there, it can corrode and the weight of the 1 kg standard may change. So what can happen? That can lead to the national. If 1 kg is national standard changing, then that effect will transfer to all the chain or the even consumer also. So we'll see this thing also. So what happens that? So every country is having National Meteorology Institute. India is also having one National Meteorology Institute, and that is called National Physical Laboratory for India. We are maintaining the national standards or SI units in physical form. They are defined by the BIPM France. But National Physical Laboratory for India is having, like similarly US is having one NIST USA, and then one National Physical Laboratory in UK also. That is also maintaining the national standards for UK. So being National Physical Laboratory, we are having one national mandate for maintaining the national standards. At the same time, we are involved in the research also. So two things we are doing. Fine. So leave them. So at National Physical Laboratory, currently what we are doing, at 1 kg, 1 meter, so what we are currently doing, even the time which you are having in your mobile, so that is also coming from the National Physical Laboratory. Initially that was coming to US servers, but for the national for reliability and strategic operations, US time is not that much reliable. So we had to have our time. So now we are providing our time from there itself. Fine. So our agenda will be like, what this presentation will be, what we will be doing, we will be discussing about the basics of the biosensors, what kind of biosensors you are having, evolution of biosensors, and after that we will be discussing what novel we are doing, and we will be giving you an example of biosensors, how that can be used and for disease detection. Okay. So basically, biosensors are like, you might be having heard about that COVID detection kit was released when Corona was there. Rapid detection kit. Initially you used to go for laboratory, and that results used to come in 3-4 days. But later, at the end of when the Corona was going like around like 3-4 months, after 3-4 months, one kit was developed for biosensor rapid detection. And that was a kind of biosensor. The glucose monitor which that diabetic people used to have in their home, and that is also biosensor. Okay, so all these are example of biosensors. Why they are important? Because rapid detection. You don't need, like many times you need a qualitative assessment of the disease, whether disease is there or not. For detailed analysis, always you can go and consult to a pathology lab. But what happens that you need some assessment whether that is there or not. For that, and sometimes biosensors, now the technology is improved. Sometimes biosensors are so much advanced that will give you the quantitative analysis also. What percentage of disease is there? Instead of, and in addition to giving yes and no, they will be giving percentage. 60% disease is there. Like at what stage disease is? The glucose, for example, the glucose, most advanced biosensor current nowadays is glucose detection. That gives you quantitative analysis. 105, 106, that gives you glucose level in your sugar level in your body, blood serum. So what is that? An analytical device that combines biological recognition element with a physicochemical transducer. Okay? So what biosensors are having? They are having a biological recognition element with a physicochemical.
The simple idea is that when we are modeling using regression, our model looks something like this. That is a linear combination of the input variables. Alright, this is a very, very simple definition of linear regression. The linear regression is a little bit more advanced than this. Okay, so let us stick to it for now. Okay. Yeah, I'll touch a bit of it so that you will know what I am going to use in my problem. So the regression methods, there are different variants of regression methods. So if you go to the Python and then you start using these regressions, so there are various key terms that you will see there. Even in MATLAB also currently you will see. So there is pseudo inverse least square regression. Then there is backslash operator, and then we have lasso regression, robust fit, and ridge. Okay. Now all of these regressions, they have various purpose for different data set it is being used, and it gives you something result that you want in your model. What do you mean by this? So in simple words, I'll explain. Suppose you have 10 variables, but you do not want those 10 variables in your model. Okay. This means you want more sparsity. So you will go for lasso regression. Now, someone else will say, no, I want all those 10 variables in my model with small weightage. So then you will go for ridge regression. So on and so forth. All these things can be explained accordingly. Why I talked about lasso and ridge? Because I am going to use ridge regression in my problem. Okay. So how it is being this lasso, ridge, p-inverse, backslash, operator, all this is how they are being decided, and what is the mathematics behind it is very, very important. You are a mathematician, you should not talk like a computer scientist. I respect computer scientists, but they don't know the inside mathematics. So we are better, right? So what we have to do is that here we should understand this piche kya ho, p-inverse, lasso, ridge, इसमें piche ho kya hai? What is the mathematics behind it? So then you are a bit ahead of those who do not know the mathematics. And that is where the idea of SciML comes, scientific machine learning, where you know the scientific theory and then you also know the machine learning. Right? So here what happens, this is the standard regression model. What we are doing, this is basically an optimization problem. If anyone is from optimization here, there is an optimization problem. So what we are doing is that we are minimizing this function ax minus b modulus 2. So what is ax minus b? So technically we are solving this ax equals to b, and we are solving this ax equal to b for any regression problem, and then we minimize. What are we minimizing? What is this? Norm to ax minus b. What are you saying? So we are minimizing this error. Okay, whatever error would come, because we do not, when you are doing the regression many times, either you are try to find this or this or this. Okay, so we're minimizing this error ax minus modulus 2 of a minus b and plus lambda 1 non-maximum plus lambda 2 non-maximum. So this is non-maximum times lambda 1 and lambda 2 is norm of x2. Lambda 1 and lambda 2 they are known as regularization coefficients in the equation. Okay, so depending upon whether you choose lambda 1 to be 0 or lambda 2 to be 0 or both lambda 1 and lambda 2 to be 0, we have all these variants of regression techniques. Okay, why we choose lambda 1 0, why we choose lambda 2 0, I teach all these things in my class. It's long lecture. So, but you should know that based on these lambda 1 and lambda 2 values, 0, non-zero, we decide which regression to choose. P inverse कब होगा? जैसे for example, for ridge regression, we make lambda. For ridge regression, we make lambda 1 0. Ridge, yes. Lambda 1 is 0 for ridge regression. Okay? We do not want to promote sparsity because if we make lambda 2 0 and keep lambda 1, that will promote sparsity. Okay? Might have written it. Yes. For ridge regression, lambda 1 is 0. Okay? Alright, so let us come to the problem. Once you have the idea of the technique, the methodology that we are going to use. So let us come to this. Yeah, a bit of about scientific machine learning. What is scientific machine learning? So this is, if you see from here, you get some idea. We have here the machine learning techniques, and we have the scientific theory, and an amalgamation of the two gives rise to a very niche area of study in the data, that is SciML or scientific machine learning. What is the advantage of using scientific machine learning? It is powerful. Why it is powerful? Because it uses data very efficiently. So it's powerful. It is robust. Why it is robust? Because it respects all the physics of the given problem. Because we know the physics of the problem. Why you are an expert in constructing buildings, you are a civil engineer. So you know the physics of how to construct a building. Now you are using the data set efficiently to improvise your model. Then it is interpretable as well. Why is it interpretable?
Interface. Interface. Which is according to your face, if you can move your eyes, no, if you will move from left to right, it will move. It means it dragging face. If it will be better if only 1% is just out of the you have to now you can move left Basically, the reason is the multiple phase, it will choose only one phase. We have programmed it like that. And in the second of all, there was a dependent lighting condition. It is a time constant condition. So we get this very easy. Otherwise, we have also done a feature that
1 candela, 1 kg, 1 second. But that is the definition. And how we have to measure and like how that definition will be dissipated to the consumer, that has to be decided, and that has to be transported, and that has to be dissipated accurately. That distribution has to be done accurately. Okay. So being in National Metrology Institute, NMI, and that all those measurements comes in the definition under comes to the metrology basically. And being the National Metrology Institute of India, we are maintaining those SI definitions, SI units at there. So we are maintaining in physical form all the SI units, and those physical SI units are being distributed throughout the country. And that is the definition. We may not be directly connected to the consumer, but all the manufacturers, all the stakeholders are connected to us, and all the instruments which are coming to you, they are calibrated, and certainly they are having some chain of calibrations, unbroken chain of calibrations are reaching to ourselves. Okay? So this is what National Measurement Institute of India, this is here. And that's how measurements, first I told you how measurements are important for you and from where those measurements are coming. And this is one aspect of these measurements are from the point of view of consumers. But another point of view, if you are exporting something and you are saying that is 1 kg or 1 meter, but another country is saying, okay, that is only 900 grams or 0.9 meters. How will you define that? How will you defend that? Recently you might have heard about some like 3 years ago, there was some news in the media that some shipment was there from India and that was exported to US or some UK, and that whole shipment was returned back because of one measurement was not accurate according to them. And that shipment, whole rice shipment was there, and because of transportation cost was too high, and all the rice grain they have to dump all the rice grain into the sea because they were not accepting. They said it was a quality issue. But later we found out when we did all the analysis at our institute, we found out that everything was correct. It was only the issue of certification, and their certification was not matching our certification. So if they have been certified by the NMI, this issue might not have happened. Whatever measurements are conducting at here, they are worldwide accepted. And if you are exporting something which is certified by here, they will be accepting that thing, because all the key comparisons, our 1 kg, 1 meter, all the key quality comparisons are accepted worldwide. Their measurements are accepted, their instruments are coming to us for calibration or testing. We are sending our instruments to their lab for certification, calibration, testing. We are testing each other's instrument so that they are accepting, so that whenever we are certifying anything, they will be accepting, and their certification, they will be accepting our certification, and there will be no glitches or no return of all these things. So why these measurements, accurate measurements are important? Because at our domestic level, financial irregularity, the financial loss should not happen. At the same time, at the international level also, all the certification measurements are widely accepted. And that's how the measurements plays key role in our daily life. Generally, we ignore all these things, or that is inaccurate measurements are so common in our life that we say, okay, ठीक है, कोई बात नहीं, चलता है, ऐसे करके. But that is not the case.
So my question is, as you said that some assignment was cancelled. So when they were exporting it, there must be some terms and conditions.
Some terms and conditions.
They might have defined that you have to set in this measurement.
Yes. But they were not certified. They did not take any certification from anywhere.
Okay.
They did not take any certification from anything from any lab. Or the lab from which they took certificate was not competent enough to certify that. It happens. So that's why, if you are taking certification, from where you are taking certification, just like degree. You are taking degrees from anywhere, or a degree from any other institute, premier institute. So that's how they will be widely accepted. Our education certificate, some certification, online certification will not be that much accepted. But if you are taking some offline certification, offline course, that will be widely accepted.
All the faculty members who have come from outside and Jaipur, and all the other dignitaries who are here, first of all, welcome you all on this faculty development program on recent trends of mathematical and computational biology. When this idea came that we should choose this topic, then now what we feel as that the time has come for multidisciplinary approach, because here in mathematics, in CS, computational and biology, all are put together for discussion over here, and all the experts from different backgrounds came here for interaction and learning things. So what I feel is that for any kind of problem solving, just take an example of a Smart India hackathon that is in all the colleges. This time, students are solving the problems that are mentioned in their portal, and these all are problems which our industries are facing it out. So while they are making the team, they are approaching, first year students are approaching to different branches, because some mechanical thing prototype has to be made, the mechanical they are putting in their team, electrical engineers they are putting in the team, and together they are solving the problem that is related to any kind of subject. In Poornima also, we are always motivating our faculty members to continuously learn in their focus area, so that they can, students' ultimate goal is the students who can be benefit out of it. So such kind of faculty development programs are always encouraging, and we congratulate Shilpi ma'am and Nikita ma'am for successfully organizing this event in Poornima College of Engineering. And all the participants who are coming from outside, I hope they have very interactive sessions that has happened, and the eminent speakers who have come here and they have shared the knowledge that must have been benefited. You must have been benefited. So once again, welcome you, Chief Guest and Guest of Honor, and on this, because we were discussing here that he has to go, Pilli sir has to go on another engagement. I'm not taking much of time, and once again, welcome.
Thank you, sir. Now, we are delighted to invite our guest of honor, Dr.
Knowledge that Dr. Shilpi has been very instrumental and very active during the process. She has been very actively engaged, like forward this, forward that. So it was very active role. She has played very active role, and very actively organized all these things, and that shows sincerity in this approach. Now come to this technical development. And it has been a wonderful experience to meet such engaging participants, and whoever has attended this thing. And it has been a good experience for me too. And as Thimla sir has said, the world is interdisciplinary nowadays. Whenever you are going to develop, or when you are going to develop anything or going to research on anything, you should be like engaging many people in your team, including your team, or you should have certain different experience from different areas. So it is a one person cannot solve everything. Like it's a teamwork always. So you should have different flavors for different areas, and you should be knowing what to include in them. And at the same time, I wish that this experience of the participant you will be able to transfer this experience or this learning to your students also, so that and will be able to motivate them to engage in different research areas, develop product, technology transfer, because nowadays startup culture is also growing in India. So instead of looking for job, students also always have an option for startup, and at the young age they starting the things, there are chances that they will be getting success in that. So my message for students and for the teachers of this, you can motivate students for the startup culture also, and that will promote our goal of our Prime Minister, that Make in India. Because this is a really right time. People things has gone really easy for setting up a company or setting like we have got lots of infrastructure also to manufacture, to make, to innovate, and the resources are also available online. Like you can use many things, like even if you are using resources, internet is also not that much costly as it was earlier. So these things are very easy nowadays. So I wish best of luck to all of you. I will not be taking much time, and thank you once again for inviting. Thanks.
Present here. On behalf of organizing committee, it's my privilege to deliver the vote of thanks at this valedictory session of Faculty Development Program on recent trends on mathematical and computational biology, organized under the aegis of ATAL Academy and AICTE. First and foremost, I extend my heartfelt gratitude to AICTE and ATAL Academy for granting us this opportunity to host this FDP and for their continuous support in promoting the faculty development program and academic enrichment. We are deeply honored by the presence of our chief guest, Dr. Pilli, and guest of honor, Dr. Arukant sir, graced this occasion and shared their valuable insights that have inspired us. Thank you for taking the time out of your busy schedule to encourage and guide us. A special word of appreciation goes to our eminent speakers and persons who have enlightened us with their expertise on various dimensions of mathematical and computational biology. Your lectures, discussions and interactive sessions have truly broadened our horizons and given us new perspectives of teaching and research. I also take this moment to thank our management, Director Sir, our Vice Principal Sir, Dr. Pankaj Demla, management for their encouragement and unwavering support throughout the conduction of the FDP. Our sincere thanks to the participants from across the institutions who have actively engaged in the sessions with enthusiasm and curiosity. I cannot forget to acknowledge the efforts of organizing team, technical team, supporting staff and my dear students. Without your behind the scenes coordination, dedication and commitment, this event would not have been possible. Finally, I thank once again, extend my gratitude to this program, and we look forward for carrying the learnings from this FDP and applying them in our academic and research perspectives. Thank you, Madam.